In this chapter, we've learned something about the cell cycle, the growth of the cell, the preparation for division, and the division of the genetic material and the division of the cell. Today, we want to talk a little bit about how the cell regulates what it does. What are some of the things that trigger the cell to make sure it divides properly? Today, our essential question can be, how do cells know when to divide? Knowing when to divide prevents cells from dividing too early so that they do not have enough resources or genetic information to function properly, or they divide too late when the cell has been damaged because of too much waste material or not enough information to form the proper materials. In this section, we'll also talk about the chemicals and other signals that are used to tell the cell when to do its division, when to start its mitosis, when to start the copying of the genetic information. All these things are very important. Plus, we'll also talk about what causes the cell to decide to stop working and break down and be gone. When we talk about cell cycle regulation, one of the key things to think about is that proteins, which are basically the all-in-one tool that cells use to do what they need to do, become a big part of the cell cycle. And in fact, regulatory proteins, both inside and outside the cell, are the triggers that tell the cell what to do. These regulatory proteins are the controls that control the cell growth and division, and they turn the process of cell growth and division on and off as needed. An example is in this picture is when a broken bone occurs. There are cells that are stimulated to divide rapidly, and they start the healing process. They start the flow of blood to the site. They bring in the necessary cells that can regenerate the crystal portion of the bone, and the cells that will glue everything together. The rate of the cell division slows when the healing process nears completion. As the cells detect that the bone is nearly knit, then they will slow down. Otherwise, we'd have big, huge lumps where we have a broken bone. One of the key proteins in the process of cell division is called cyclins. Cyclins are a family of proteins that regulate the timing of the cell cycle in eukaryotic cells. This graph shows how cyclin levels change throughout the cell cycle in a fertilized clam egg. If you look at the graph, you will see that it has an increase in cyclin content, and then it decreases rapidly, and then it slowly increases, decreases rapidly, slowly increases. And those correspond to mitosis and interphase, and then mitosis and interphase. So when we look at how this material regulates the cell, it tells it when to do the division. Regulatory proteins do a variety of things. They can come both from the outside and the inside of the cell. Internal regulators are proteins that respond to events inside a cell. They allow the cell cycle to proceed only once certain processes have happened inside the cell. In other words, the cell can't divide until mitosis is complete. So if we're caught in one of the phases, like anaphase, before it's ready to divide, the cell will not divide until anaphase is complete. There are also external regulators that are proteins that respond to events outside the cell. They direct the cell to speed up or slow down the cell cycle. Other regulatory proteins are called growth factors. And they're external regulators that stimulate the growth and division of cells. They are important during embryonic development and wound healing. Cell growth and division are important, but once a cell starts to have problems, once the DNA is damaged or a portion of the cell is damaged, something has to happen so that cell cannot continue on and cause further problems. The process that we talk about is called apoptosis, which is a process of programmed cell death. Apoptosis plays a role in the development by shaping the structure of tissues and organs in plants and animals. For example, the foot of a mouse is shaped the way it is partly because the toes undergo apoptosis during tissue development. When we talk about controlling the cell growth and the cycle of the cell, one of the key things we need to talk about is something called cancer, which is considered to be an uncontrolled cell growth. And we need to understand how cancer cells differ from other cells. When something happens inside the cell and the damage to the cell itself and to its nucleus, the cancer cells do not respond to the signals that regulate the growth of the most cells. As a result, the cells divide uncontrollably. They go out of control. They start using a lot of the body's resources, and it also starts to interfere with other organs and cells that aren't associated with the cancer. Cancer is a disorder in which body cells lose the ability to control cell growth. Cancer cells divide uncontrollably to form a mass of cells called a tumor. Cancer cells are not always a bad thing. They're not always a good thing, but they're not always a bad thing. A benign tumor is non-cancerous. It does not spread to the surrounding tissues. It's just a growth that occurs, and many of us have small, what we call malignant or benign tumors. A malignant tumor is cancerous. It invades and destroys surrounding healthy tissues and can spread to other parts of the body. The spread of cancer cells is called metastasis. Cancer cells absorb nutrients needed by other cells, block nerve connections, and prevent organs from functioning. Cancers are caused by defects in genes that regulate cell growth and division. Some sources of gene defects are smoking tobacco, radiation exposure, defective genes, and viral infections. For instance, a damaged or defective p53 gene is common in cancer cells. It causes cells to lose the information needed to respond to the growth signals. When we talk about treating cancers, sometimes for localized tumors, we remove it by surgery. Many tumors can be treated by using targeted radiation. Others will be treated with chemotherapy, which uses compounds that kill or slow the growth of cancer cells. More often than not, we usually use a combination of two or all three of them to treat certain types of cancer. How they are chosen depends upon the genome of the cancer and also on the type of cancer that they see.